Hi, I'm Alyssa Nelson. My group is going to be discussing brain imaging methods in this presentation. Neuroscience research involves studying the nervous system and exploring its development, structure, and function. There are many branches of the discipline, and each has its own specific goals and areas of focus. Regardless of specialization, neuroscience researchers ultimately use the same imaging technology to learn more about the brain. In this lesson, we are going to explore some specific types of brain scanning equipment that are most frequently used. The first is CT scanning. A CT scan combines X-rays of cross-sections of the brain, which creates a 3D image of the brain. It provides more detailed information about brain structures and tissues in regular X-rays, and it helps localize brain injuries and disease. It also helps researchers map brain regions associated with different behaviors, often by studying people with specific brain injuries. Its discrimination time is less than 1 millisecond, which is why it is often used in acute emergencies compared to MRIs, which have significantly longer imaging time. Another strength of CT is that it has excellent spatial resolution, which enables it to differentiate between structures that are close together. However, the contrast resolution of CT is inferior to other imaging modalities such as MRI. This can be attenuated by administering IV contrast material. A primary disadvantage of the CT scan is that it only shows static structures and doesn't reflect dynamic changes. Thus, unlike PET imaging or fMRI, it does not provide information about brain activity and function. For this reason, it is not used very frequently in contemporary neuroscience research.
An EEG, or electroencephalogram, is a method of measuring brain activity through measuring the electrical activity of the brain. An EEG is conducted by attaching many small metal electrodes to the scalp of the patient. These electrodes are attached to wires, which then feed into the computer of the EEG machine. These electrodes can track the activity of the electrical impulses in the brain, which then are displayed on a computer screen in a wave graph that the doctors administering the EEG can read. While the test is administered, the subject is asked to lie still and are asked to close their eyes. Different from other brain scans, where the patient might be asked to look at images or perform tasks, the only tasks potentially required of the patient during an EEG include changes in breathing, sleeping, and looking at flashes of light. The conditions that an EEG is most commonly used to diagnose and monitor are lesions of the brain, strokes, sleep disorders, seizures, and Alzheimer's, as well as other conditions. The doctor administering the test can interpret the results of the scan to make the proper diagnosis and recommendations for the patient's conditions.
Functional magnetic resonance imaging, or fMRI, was first developed and used in 1990. Since then, it has been used to map and understand many cognitive, motor, and sensory functions in the brain. The basis for fMRI is blood oxygen level dependent, or BOLD signaling. BOLD contrast relies on changes in the magnetic fields that surround red blood cells in the brain. These changes are caused by shifts in hemoglobin oxygen levels. When an fMRI scanner exposes the brain to different magnetic fields, hydrogen protons act like tiny magnets that indicate oxygen level changes. fMRI has a very high spatial resolution, but its temporal resolution is lower than many other measures of brain activity. Spatial resolution refers to the location accuracy of measured brain activity, whereas temporal resolution refers to the accuracy and timing of that measured activity. fMRI is used both to learn about healthy brain function and to explore how function is impaired by diseases and disorders, largely because it can detect structural abnormalities that many other imaging techniques can't. Many diseases have been studied using fMRI data, and shown here are examples of fMRI images comparing brain activity of people with Alzheimer's and alcohol addiction to control subjects. Beyond descriptive studies of normal and disordered brain function, fMRI is used in experiments, surgical planning, and to monitor treatment outcomes.
The positron emission tomography scan, also known as the PET scan for short, is similar to the fMRI in that it measures metabolic activity in the brain. The PET scan measures metabolic activity by tracking a radioactive ligand. This ligand can be a neurotransmitter or other suitable substance, but usually researchers use radioactive glucose, such as the one shown on the slide. By tracking the radioactive ligand, the PET scan can detect which areas of the brain the ligand is absorbed into, which indirectly measures neurological activity. Since researchers can visualize what areas of the brain are active, just like in fMRI, the PET scan provides great spatial resolution, but it has poor temporal resolution for the same reason as fMRI. Since PET scans provide similar information as fMRIs, they can be used for the same types of studies. There are advantages and disadvantages to both types of scans. PET scans do not have as good resolution as fMRIs, as you can observe in the images on the slide. fMRIs, however, may have trouble imaging certain areas of the brain because those areas are surrounded by tissue that have different magnetic properties than the brain tissue. There are additional differences that can be continued to be discussed, but in general, it is ultimately up to the researcher to decide what imaging technology is best suited for this study. This concludes our lesson on common brain imaging technology that neuroscientists use in their studies. Hopefully this lesson was helpful and you learned something. Thanks for watching.